Hello dear students, how are you? Hope you will be good and fine. I'm Dr. Nabil, and from today we will start a series of lecture on microbiology, and hope these videos will be helpful for your medical careers. Okay guys, so first we will start from the definition of microbiology. That what is microbiology? Uh, Microbiology. It is the branch of medical science or medical biology. It is the branch of medical biology which deal with the study of microorganisms under microscope is known as microbiology. And this study of the microorganisms will be regarding to their structure, function, relationship with the other microorganisms and relationship with the environment. Deals with the study of microorganisms under microscope. And this study will be on the basis of structure of microorganisms, on the basis of function of microorganism, on the basis of relationship of microorganisms with other microorganism relationship of the microorganisms with higher organisms and the relationship of microorganisms with the environment. All this study is known as microbiology. Okay, now we are giving quick review to the definition of microbiology. That it is the branch of medical biology deals with the study of microorganisms under microscope on the basis of how you will study the organisms under microscope on the basis of their structure, function, relationship with other microorganisms, relationship with of microorganism with higher organism, and relationship of the microorganisms with the environment. All this study is known as mean all this study, such type of study of microorganisms is known as microbiology. Now, what we are going to study in this field? Organisms. Now, types of organisms. Organisms. So there are two types of organisms. One is called macroorganism, and the other one is called microorganisms. Now, in the previous classes we discussed about that macroorganisms are those organisms which are visible to the naked eye. We didn't discuss about any size or anything else. And micro are those organisms that which can't be seen by the naked eye. Yes, but as you came to the higher classes, now your definition should be more precise. So the difference between microorganism and macroorganism, it depend on their size. So the difference between microorganism and macroorganism, it depend on their size. It depends on their size. Now, a healthy eye will be able to visualize all those objects whose size will be up to 200 micron or more than 200 micron. So, a human healthy eye is able to see only those objects whose size is up to 200, yeah, more than 200 micron. So what is the size now here? It is 200 micron. So now, if the size of an organism is less than 200 micron, so a healthy eye will be not able to visualize them. So a healthy eye will be not able to see these objects. If the size of an object is less than 200 micron, so a healthy eye will be not able to visualize those objects. A healthy eye in this moment, this healthy eye needs an external eyepiece. External eyepiece. And that external eyepiece will be either in the form of magnifying glass, or it will be in the form of microscope. Okay, except magnifying glass use, or except the uses of microscope, we will be not able to see them. So microorganisms are those organisms whose size will be less than 200 micron, which will be not able to the naked eye, and the human healthy eye needs to visualize those objects by using of any magnifying glass, or by using of any complex structure of magnifying glasses, which is known as microscope. Clear? Now, about macroorganisms. Macroorganisms are those organisms whose size will be approximately 200 micron or more than 200 micron. If the size of an object, yeah, organism is 200 or more than 200 micron, so a healthy eye will be able to visualize those objects. And this eye don't need any external eyepiece, no need of any external eyepiece. Clear? Now we are going to discuss about the grouping of organism. Grouping of organisms. All the organisms, initially all the organisms were classified into two main groups, and those were classified on the basis of their cellular structure and on the basis of their uh, now we are going to discuss about classification of organism. Initially all the organisms were classified into two main groups, that is known as plant kingdom and animal kingdom. And all the organisms were placed in the plant kingdom, either animal kingdom, and this classification was based on the features of organisms, on the cellular feature of organism. So let's see what were the features on the basis of which they were distributed or they were separated from each other and placed in different groups. Animal kingdom. In the animal kingdom, all those organisms were placed who lacks cell wall as their outermost layer. In the animal kingdom, all those organisms were placed who lacks are absent cell wall as their outermost layer. Next, all those organisms were placed in the animal kingdom uh, who lacks chlorophyll. Chlorophyll in their structure. If they don't have chlorophyll, so there will be no process of photosynthesis. Synthesis. And if the process of photosynthesis is absent in animal kingdom cells, so these cells will be not able to produce food for their own, mean energy will be not produced by their own, can't produce energy by their own. By their own. Clear? Next, they will then the nucleus will be at the center. Center of the cell. Cell. Next, they will carry small contractile vacuoles. Vacuoles, etc. Other features are also present. Okay. Now, let's see about plant kingdom. In the plant kingdom, all those organisms were placed who possess chlorophyll, they possess cell wall, have cell wall, and they have chlorophyll. If the chlorophyll is present, so they can the process of photosynthesis will be present. Synthesis. Next, they can be able to produce food, yeah, energy, to produce energy. Next, the nucleus of the plant kingdom cells will be not at the center, but it will be pushed to one side. Nucleus will be at one side. At one side. It will be pushed one side. Why? Because they will carry one large contractile vacuole. Not contractile, one large vacuole. And due to that vacuole, the nucleus will be pushed with one side. Okay. Now, with the passage of time, with the passage of time, new organisms were introduced in the field of microbiology. New organisms introduced were introduced in the field of microbiology. Microbiology, whose characteristics were character wise, whose characteristics were same same like like animal kingdom and similar to the and also to plant kingdom organism, and some new other organisms were also introduced in the field of microbiology, whose characteristics were not similar to the animal kingdom, neither neither plant kingdom characteristics. Kingdom. So a new problem were created. Now With the passage of time, some new organisms were introduced in the field of microbiology whose characteristics were similar to plant kingdom as well as to animal kingdom. But meanwhile, some other organisms were also introduced whose characteristics were not similar to the plant kingdom, not animal kingdom. So a problem was created once again. A new problem was established, and in the and in 1866, Ernst Haeckel, Ernst Haeckel, Haeckels introduced a new kingdom which was named as Kingdom Protesta. And in the Kingdom Protesta, the organisms were placed on the basis of their in the Kingdom Protesta, the organisms were placed on the basis of their nucleus, structure of nucleus, as well as on the basis of their the cellular organelles. Organelles. Further, the Kingdom Protesta were classified into two groups. It is further classified into two groups: prokaryotic group and eukaryotes. Prokaryotes and eukaryotes. The Kingdom Protesta were classified into prokaryotes and eukaryotes. And in the prokaryotes, all those organisms were placed which are carrying primitive nucleus. Primitive means that the nucleus of these group of organisms will be not mature, immature nucleus. Immature nucleus. What is primitive nucleus? That nucleus is known as primitive nucleus whose genetic material, chromosomes, will be not enclosed within the nuclear membrane. Okay? Or DNA will be not enclosed within the nuclear membrane. While in the eukaryotes, all those organisms were placed which possess true nucleus. Clear? Now we will discuss about the difference between prokaryotes and eukaryotes. Now let's discuss about the difference between prokaryotes and eukaryotes. Prokaryotes and eukaryotes. Now we will briefly discuss about it. So we will start from their size. That what is the size of prokaryotes and what is the size of eukaryotes. So we are going to start from their size. In the prokaryotes, all those organisms were placed whose size is less than 5 micron. And in the eukaryotes, all those organisms are placed whose size is more than 5 micron. Okay. Now let's start from the nucleus of the prokaryotes and eukaryotes. Nucleus. The nucleus of the prokaryotes is primitive nucleus. Primitive nucleus. While the nucleus carried by the eukaryotes is true nucleus. True nucleus. Nucleolus. The nucleolus is absent. Okay, then now what is primitive and what is true nucleus? Okay, primitive nucleus is that nucleus whose genetic material will be not enclosed in the nuclear membrane. So let's discuss about nuclear membrane. The nuclear membrane is absent in the prokaryotes, while the nuclear membrane is carried by the eukaryotic cells. And within the nucleus of the the genetic material will be enclosed. Okay. Now nucleolus. Nucleolus. The nucleolus will be absent in the prokaryotic cells while it is carried by the eukaryotic cells. Clear? Now, deoxyribonucleoprotein. Deoxyribonucleoprotein. The deoxyribonucleoprotein are absent in the prokaryotic cells while they are present in the eukaryotic cells. Clear? Now, let's discuss about chromosomes. The chromosome present in the prokaryotic cell is single and circular type. Circular mean it will be of such type. It is single and circular, while the chromosome present in the eukaryote is one. It will be one or more than one, and the chromosomes will be linear type. Linear type mean such type of chromosomes will be carried by the nucleus of eukaryotic cells. Clear? Now let's discuss about the mitotic division which will be present in the eukaryote. Mitotic division. The mitotic division is absent in the prokaryotic cells. Absent in the prokaryotic cell, while the mitotic division is present in the eukaryotic cells. How the prokaryotic cells will divide their cells? By binary fission. By the process of binary fission, prokaryotic cells will multiply and divide their cells. Okay. Now, Golgi apparatus. Golgi bodies. The Golgi bodies are absent in the prokaryotic cells while they are present in the eukaryotic cells. Next, ribosomes. Ribosomes. The ribosome present in the prokaryotes is 70S type of ribosome, and they have larger subunit and smaller subunit. The larger subunit consists of 50S and the smaller subunit consists of 30S, while the ribosome present in the eukaryotic cell is 80S, and they are consist of two subunit, larger subunit and smaller subunit. The larger subunit consists of 60S and the smaller subunit consists of 40S. Okay. S means Swedberg unit, or it is also known as sedimentation coefficient, or it is also known as sedimentation rate. Clear? So don't be confused among these. Now let's discuss about endoplasmic reticulum. The endoplasmic reticulum are of two types, rough and smooth. Both type of endoplasmic reticulum are absent in the prokaryotes, while those will be present in the eukaryotic cells. Clear? Now let's discuss about another granular structure which is known as lysosome. Lysosome. The lysosome are absent in the prokaryotic cells, while they are carried by the eukaryotic cells. Clear? Now let's discuss about the sterol which is present in the cell membrane. Sterol. The sterol are absent in the prokaryotic cells, while they are carried by the eukaryotic cell. And what is the function of sterol? It is giving the elasticity to the skin, it is giving elasticity to the cell membrane and prevent the cell from the dehydration. Okay. Now cell wall. Cell wall. The cell wall of the prokaryotic cell is complex type. While the cell wall of the eukaryotes is simple type. Clear? The cell wall of prokaryotes carrying N-acetylmuramic acid. Okay. Now respiration process of respiration. The respiration process in the prokaryotes is done by mesosome. And mesosomes are of two types, lateral mesosome and septal mesosome. Especially lateral mesosome is responsible for the respiration process, while septal mesosome, it is responsible during cell division. It is going to divide first the genetic material of the prokaryotic cell and then helps in the binary fission. Okay, division time septal mesosome is helping, while energy production time the only lateral mesosome is helping in the production of ATP. Okay, and respiration process occur in the eukaryotes. It is done by the mitochondria. And mitochondria is absent in the prokaryotic cells, while mitochondria is present in the eukaryotic cells. Clear? Now, example of the prokaryotes and example of the eukaryotic cells. The example of prokaryotic cell is bacteria, and blue-green algae. Algae. While the example of eukaryotes is algae, except blue-green algae. Algae. Only blue-green algae is the example of prokaryote. Rest of other algae are coming in the eukaryotic organisms. Protozoa, fungus, etc. These are the example of prokaryotes and eukaryotes. Now, let's discuss about the effect of microorganisms. Effects of organisms, microorganism especially. So, the effect of microorganism is classified. They have two types of effect. Two types of effect. They are good effect. They have good effects, or it is also known as beneficial effects, and they have bad effect, which is also known as harmful effect. Bad effects, or it is also known as harmful effects. Now, let's discuss about good effect of the microorganism. So, they helps in the formation of wine and alcohol. They helps in the formation of wine as well as in the formation of alcohol. Clear? Next, they also helps in the formation of citric acid, acetic acid, etc. They also helps in the formation of vitamin and antibiotics. Which are used for the treatment of those diseases which is caused by the microorganisms. They also helps in the formation of yogurt, and yeast. They also helps in the formation of yogurt as well as yeast. They also helps in the formation of okay, some bacteria are methanogenic, which are responsible for the production of methane gas. And methane gas are also known as biofuel. Okay, these are the good effect of microorganisms. Okay, now harmful effect of the microorganism. As the microorganisms have good effect, and next good effect of the microorganisms that they increase the soil fertility. They helps in increasement of soil fertility. Means some bacteria are converting the atmospheric nitric oxide into nitrate, and the nitrate is responsible for the increasement of soil fertility. Clear? Now, as the microorganism have good effect as well as they will also have bad effect. What are the bad effect of microorganisms? That they are going to spoil food and water. And water. Okay, and if a person or an organism eat spoil food or drink spoil water, so it will be lead to the disease. It can causes disease. Causes diseases. Diseases. Causes diseases in animal as well as plant. Plants. They also spoiling our leather jackets and shoes. And shoes. They also spoiling our house paint. Paint. They also spoiling the pipeline which is used for the supplying of water. They also spoiling our clothes. They also spoiling our in short, they are going to spoil our furniture. Furniture. And in short, they are going to disturb our lifestyle. Lifestyle is going to be disturbed. Clear? Now, let's discuss about the branches of microbiology or medical microbiology. Branches of medical microbiology. So, the medical microbiology have branches which consist of there are okay, the first one is general microbiology. Second one branch is bacteriology. Third one branch is virology. Fourth one is mycology. Next one branch is parasitology. Next one branch of the medical biology is immunology. Next one branch of the medical biology is clinical microbiology. Clinical microbiology. Okay guys, now who, if someone is asking from you that who is the founder of modern microbiology, so you can answer that Antonio van Leeuwenhoek. Antonio van Leeuwenhoek. Okay, Anthony Leeuwenhoek is the founder of modern microbiology. Clear? And that's the end of our first lecture. Hope students, hope you will enjoy it, and hope so that you will get some helps in the microbiology. Thank you.